the radiant energy, utilizing the radiant energy and producing energy-rich products at the end. The energy-rich products that are produced at the end of photosynthesis are nothing but the glucose or the starch, etc. So now this photosynthesis is restricted only to certain group of organisms, that is plants, mostly green plants having chlorophyll or able to do this photosynthetic activity. But the other important phenomenon that is associated with all living organisms is respiration. There is no organism as such without performing any respiration. Many organisms, of course, only plants are able to do photosynthesis, while respiration is the important biochemical reaction that is associated with all organisms. In all living beings, they exhibit what we call growth, reproduction, respiration. Of course, the food production is found in all organisms, but only autotrophs are capable of converting this chemical, light energy into chemical energy. That is photosynthesis. Now, this phenomenon of respiration is found in all organisms, including human being. If our respiration stops, means we are no more alive. All living organisms do respire. Respiration is the fundamental phenomenon associated with all organisms. Okay, so now this antagonistic to photosynthesis, which is an anabolic process, the respiration is a catabolic process. We have seen in photosynthesis, carbon dioxide combines carbon dioxide in the presence of water by chlorophyll molecule trapping light energy, converts into C6H12O6 plus 6O2 plus 6H2. So the end product of photosynthesis is C6H12O6, that is glucose. Glucose, of course, through various modifications, it ultimately changes to starch. So now this energy-rich product will be utilized by all organisms, breaking the bonds of these complex molecules and converting them into simpler molecules. When these bonds are broken, energy is released. That energy will be utilized for organisms for various life activities such as growth, development, respiration, for all processes, reproduction. So now this respiration is just opposite to photosynthesis. In respiration, for example, the balanced equation of respiration is, say, for example, 6 C6H12O6 plus 6O2 changes to what we call 6O2, that is in the presence of oxygen. You get 6 CO2 plus 6 H2O plus 686 kilocalories of energy. This is a balanced equation of respiration. Okay, but this respiration, by looking at the equation, I told you C6H12O6 plus 6O2 I said. That is, in respiration, what is happening? Organism is taking in oxygen. So in the presence of oxygen, the glucose molecule, energy-rich glucose molecule is broken into carbon dioxide, water, and energy. This energy is equivalent to 686 kilocalories. But of course, in all organisms, this amount of 686 calories of energy may not be available. This is possible only in organisms that do respiration by taking oxygen. So the organisms that respire utilizing oxygen are the organisms that break glucose are organisms that burn glucose in the presence of oxygen are called aerobes. And that respiration is called aerobic respiration. There is another type of respiration wherein some organisms do not use oxygen. Even in the absence of oxygen also, they meet their environment, their energy requirement. Such organisms which do respiration in the absence of oxygen are called anaerobes. And the respiration is called anaerobic respiration. So most of the bacteria, most of the soil bacteria, and even in our body cells also, there are many, for example, muscle cells, etc. They do respire. When oxygen is not available, in extreme emergency cases, our cells are capable of doing, anaerobic respiration. Thereby, they meet energy requirements to certain extent. See, generally in aerobic respiration, the entire energy that is locked in glucose gets liberated. Whereas in anaerobic respiration, only there is a partial oxidation. There is a partial oxidation of glucose and only little amount of energy is released. So based on the presence or absence of oxygen, the respiration is of two types: aerobic respiration and anaerobic respiration. Of course, this is basically now respiration. See, is a complex phenomenon. Is a complex phenomenon. All the enzymes needed for respiration are present in a cell. Each and every cell is capable of doing respiration. Each and every cell is capable of doing respiration. But this respiration, photosynthesis, the important organelle is chloroplast. And the organelle that is associated with respiration is mitochondria. Mitochondrion is the organelle that is associated with respiration. And as the organisms do, as the cells respire, a lot of energy is released. As mitochondrion is involved in the liberation of energy, hence mitochondria are rightly called as powerhouses of the cell because they are associated with the release of energy. Hence, they are called powerhouses of the cell. If you look at the structure of mitochondria, mitochondria like chloroplast is a double membrane cell organelle, and of course, it is also like chloroplast, a semi-autonomous organelle because it has its own DNA, mitochondrial DNA, what we call. It has its own ribosomes. To certain extent, it is also a semi-autonomous organelle. Now, in the structure of mitochondria, the outer membrane is smooth, whereas inner membrane makes an entry, finger-like entries deep into the mitochondrial body, and those finger-like processes are called cristae. And the amorphous, energy-rich aqueous substance that is present inside this inner membrane is called a matrix. So, in mitochondrion, you find outer membrane, inner membrane, and on the inner membrane, you find what we call cristae. Even on the cristae, you find certain pinhead-like structures called F1 particles, are called elementary particles. Now, where are these, mitochondrion? Mitochondrion is present in a cell. Now, this breakdown of glucose occurs in a phase-wise manner. It occurs in a phase-wise manner, and for our convenience, we divide this entire process of respiration into three phases. The first phase is called glycolysis. Second is called Krebs cycle, and the third one is called electron transport system. These three are very, very important. So first one, as I mentioned, is glycolysis. This, say, you have to make it a point. The glycolysis is common for all organisms. In all organisms, whether it is an anaerobe or an aerobe, the glycolysis is common. The end product of glycolysis is pyruvic acid. So glycolysis is nothing but breakdown of glucose or breakdown of glycogen. Actually, breakdown of glycogen is called glycolysis. Or for our convenience, we start with glucose. So glucose, a 6-carbon compound, breaks in the absence of oxygen. Make it a point. So for the breakdown of glucose, no oxygen is needed. And the enzyme systems or enzymes that are involved in the breakdown of glucose are present in the cytoplasm or in the cytosol. So using the various enzymes present in the cytosol, the glucose breaks, a 6-carbon glucose molecule breaks into 2 3-carbon molecules called pyruvic acid. So glucose breaks into 2 molecules of 3-carbon pyruvic acid. So this breakdown of glucose into pyruvic acid we call glycolysis. So at the end of glycolysis, what we are getting? The end product of glycolysis is a 3-carbon pyruvic acid. This pyruvic acid, based on the presence or absence of oxygen, based on the presence or absence of oxygen, it further undergoes degradation. Further degradation occurs. Now, for example, in the presence of oxygen, this pyruvic acid that is produced in the cytosol makes an entry into mitochondrion, especially in mitochondrial matrix. In the mitochondrial matrix, further degradation of pyruvic acid takes place. So, pyruvic acid has 3 carbons. Now, these 3-carbon pyruvic acid breaks, and ultimately 3 molecules of carbon dioxide are liberated at different phases. So ultimately, the pyruvic acid decarboxylates. Decarboxylation means pyruvic acid loses its carbon at 3 stages, at 3 reactions. So in first reaction, when it loses 1 carbon dioxide, it becomes a 2-carbon compound called acetyl-CoA. Acetyl-CoA has 2 carbons. This acetyl-CoA at 2 places further gets decarboxylated. So thereby, all the 3 carbons get liberated as carbon dioxide. When this happens, not only decarboxylation, but dehydrogenation also occurs. When dehydrogenation occurs, a couple of protons, a couple of electrons are liberated, and these liberated electrons and protons are used in the reduction of coenzyme. So coenzyme 2, what we call NAD. NAD, by taking protons and electrons, gets itself converted into NADH2. FADH, another coenzyme, also takes in electrons and protons and ultimately forms FADH2. So the NADH2, FADH2, the reduced enzymes, what we call. The reduced enzymes. So at the end of, at the end of Krebs cycle, we are getting reduced coenzymes. These so the energy is locked in the reduced coenzymes. So the energy that is trapped, locked in the coenzymes gets liberated when these reduced coenzymes pass their electrons and protons, or especially when the electrons are passed through various electron carriers. Various electron carriers So, so there was a disconnection. Sorry for the interruption. So, when pyruvic acid enters into Krebs cycle, as I told you, at 3 places, decarboxylation and dehydrogenation occurs. As a result of that, coenzymes, reduced coenzymes of NADH2 and ATP, FADH2 are produced. And these reduced coenzymes have electrons and protons, and a lot of energy is stored in them. So in order to liberate energy from these reduced coenzymes, these coenzymes are to be oxidized. So this oxidation of reduced coenzymes, that is NADH2s and FADH2s, occurs when the electrons pass through various electron carriers. So this is what we call movement of electrons from NADH2 and FADH2 through various multi-enzyme electron carrier systems. What we call, there are 4 multi-enzyme complexes. One is what we call complex 1, complex 2, complex 3, and complex 4. There are 4 multi-enzyme complexes having cofactors in them and prosthetic groups, and the enzymes from NADH2, FADH2, starting with NADH2, they make entry into complex 1, then they go to complex 3, then they go to complex 4, and ultimately from complex 4, the electrons are passed to elemental oxygen. So when electrons pass from complex 1, 3, 4, when for each couple of electrons that pass, 4 protons are released from where all these reactions are taking place. These reactions are taking place on the cristae part. See, Krebs cycle takes place in matrix. I told you glycolysis occurs in cytosol. From cytosol, the end product of glycolysis, that is pyruvic acid, enters into matrix part of mitochondria. So here the decarboxylation and dehydrogenation occurs, and ultimately reduced coenzymes, that is FADH2 and NADH2, are passed, and these oxidation of these reduced enzymes takes place on the cristae part, on the inner membrane. On the inner membrane, you have 4, ultimately 5 complexes are there. One is complex 1, complex 2, complex 3, complex 4, and the final complex is what we call ATP synthase. And in between these two, we have in between 1 and 3, we have quinone, ubiquinone, a mobile carrier, a mobile electron carrier. And from 3 to 4, we have another cytochrome electron carrier. Cytochromes, generally cytochrome A, cytochrome A3 are the final ones. They are the final, electron acceptors. And from them, the electrons are ultimately given to, molecular oxygen. So when all these take place, when electrons move from complex 1, 3, 4, and ultimately to oxygen, the protons are pumped from matrix to the mitochondrial place, that is in between 2 membranes, intermitochondrial place. So when the electrons make a movement, for each electron, 3 protons are pumped from matrix to the intermitochondrial place. So when all this occurs, a lot of concentration of protons occurs in the intermitochondrial place. So the intermitochondrial proton concentration is more than 1,000 times when compared to the proton concentration in the matrix. So the protons try to escape. So always they try to escape, but the inner membrane is impervious. It does not allow. So there is one mechanism which is very similar to our, what we have read in photosynthesis, photophosphorylation. So here also there is a mechanism for the movement of the protons down their concentration gradient. So this is facilitated by what we call elementary particles. The elementary particles are otherwise called F1 particles. The F1 particles have what we call a stalk and a head. The head is located towards matrix and the stalk is on the inner membrane. So the energized protons move through the ATP synthase. For the movement of 3 protons through ATP synthase generates 1 ATP. So like that, for the synthesis of 1 ATP, 3 protons are needed. So the energy that is present in the protons is utilized in the synthesis of ATP. This we call phosphorylation. See, the phosphorylation in light reaction, we call it as photophosphorylation. But here, we call this phosphorylation as oxidative phosphorylation. Oxidative. So here, oxidation of these reduced coenzymes is taking place, and ultimately, the electrons are transferred to elemental oxygen. Hence, we call this as oxidative phosphorylation, wherein the movement of 3 protons through ATP synthase is sufficient for the synthesis of 1 ATP molecule. Thus, the ATP is produced. So, starting from glucose till, so glucose, first glycolysis, then Krebs cycle, ultimately, third phase is electron transport system. So at the end of electron transport system, ATP is produced, and this ATP will be utilized for various metabolic activities. This is aerobic respiration. This is what we call an aerobic respiration. So quickly, I'll explain briefly about each one. Say first of all, glycolysis. So this, as I already told you, this occurs in cytosol. Now this is a 10-step reaction, and most of the reactions are, each one is, regulated by a given enzyme, and most of the reactions of glycolysis are reversible reactions. They are reversible. So we'll begin with glucose. Glucose, taking 1 ATP molecule, becomes glucose 6-phosphate. In the second step, glucose 6-phosphate becomes fructose 6-phosphate in the presence of an isomerase enzyme. Then fructose 6-phosphate, by taking 1 ATP, it generates, or it becomes fructose 1,6-bisphosphate. The fructose 1,6-bisphosphate in the presence of an enzyme aldolase, it breaks to phosphoglyceraldehyde and dihydroxyacetone phosphate. These two are interconvertible by an enzyme triose phosphate isomerase. Now, 3-phosphoglyceraldehyde, by taking 1 ATP, one inorganic phosphate, it gives rise to 3-phosphoglyceraldehyde, gives rise to 1,3-bisphosphoglyceric acid. Here, first dehydrogenation takes place. With this, 1 NAD becomes 1 NADH2. Now then, 1,3-bisphosphoglyceric acid changes to 3-phosphoglyceric acid, and here 1 ADP gives rise to 1 ATP. Then 3-phosphoglyceric acid in the presence of mutase gives rise to 2-phosphoglyceric acid, and 2-phosphoglyceric acid changes to phosphoenol pyruvic acid, and phosphoenol pyruvic acid in the presence of an exokinase enzyme converts ADP into ATP, and ultimately we get pyruvic acid. So like this, starting from glucose to pyruvic acid, we get 4 ATPs and 2 NADH2s. But of the 4 ATPs, 2 ATPs are consumed. Hence, the net yield is 2 ATPs and 2 NADH2s. Now, when these ATPs, 2 ATPs are, of course, readily available, when these 2 NADH2s, when they enter into electron transport system, they yield 6 ATPs. So, 6 ATPs, but these 6 ATPs, recent research revealed that the NADH2s that are produced in glycolysis make an entry, not directly in the electron transport. Hence, they are capable of outside entry, what we call. Through them, we get only 2 ATPs. Thus, from 2 NADH2s, we get 4 ATPs and we get 2 ATPs. Thus, total net yield is 6 ATPs only. Now, in the second step, that is what we call Krebs cycle. In the Krebs cycle, before Krebs cycle, there is one more step, what we call oxidative decarboxylation of pyruvic acid. Here, what happens, see, the pyruvic acid, which is a 3-carbon compound, in the presence of a multi-complex enzyme, and in the presence of what we call 5 cofactors, the pyruvic acid changes to. So the pyruvic acid plus coenzyme A plus TPP, that is thiamine pyrophosphate, lipoic acid, and magnesium ions plus NAD. So this is a multi-enzyme, multi-factor complex. This pyruvic acid in the oxidative decarboxylation gives rise to what we call acetyl coenzyme A. So here there is a decarboxylation and dehydrogenation. With decarboxylation pyruvic acid, you get only 2-carbon acetyl coenzyme A and 1 NADH2. So now this 2-carbon acetyl coenzyme A enters into Krebs cycle. Why it is known as Krebs cycle? It was Hans Krebs who elucidated all the reactions in detail. He was an English biochemist who elucidated all the reactions that occur in Krebs cycle. Now, in Krebs, this glycolysis, all these reactions, these 10 reactions were in detail worked out by 3 German scientists by name Emden, Meyerhoff, and Parnas. Hence, glycolysis is also called as EMP pathway. So, EMP pathway or glycolysis are one and the same. So, here, this Krebs cycle, as it is named after in the honor of Krebs. This is also, as the first compound is a 6-carbon citric acid. Hence, it is also called a citric acid cycle. As citric acid has 3 carboxylic acids, it is also called tricarboxylic acid cycle. So this here we will see one by one. Here the acetyl coenzyme A combines with oxaloacetic acid, a 4-carbon compound, and you get citric acid. This citric acid gives rise to cis-aconitic acid. From cis-aconitic acid, you get isocitric acid. From isocitric acid, first dehydrogenation occurs and a decarboxylation occurs. So the citric acid, isocitric acid are 6-carbon compounds. By losing 1 carbon dioxide, you get 5-carbon alpha ketoglutaric acid. And also there is a synthesis of one reduced coenzyme, that is NADH2. Now alpha ketoglutaric acid in the next step, in the presence of alpha ketoglutaric dehydrogenase and decarboxylase, it loses another carbon dioxide and gives rise to succinyl coenzyme A 4-carbon compound. And here also one more NADH2 is produced. Now in the second next step, the succinyl coenzyme A becomes succinic acid. Here one GDP gets converted into one GTP. Now succinic acid changes to fumaric acid. Here from succinic acid to fumaric acid, it is not NAD but FAD becomes FADH2. Now fumaric acid changes to malic acid. Now the malic acid ultimately gives rise to oxaloacetic acid, a 4-carbon compound, and here also the final dehydrogenation takes place. malic dehydrogenase, and this yields what we call NAD to NADH2. So altogether, if you look at starting from pyruvic acid, you will see that 4 NADH2s and one FADH is created by one pyruvic acid. So we get 2 pyruvic acid, So hence, if you take these 2 pyruvic acids into consideration, you see that in all, 4 NADH2s and 2 FADH2s and 2 GTPs are produced at the end of pyruvic acid. Now these coenzymes ultimately make an entry into electron transport system. So here what happens in electron transport system? The electron from NADH2 and FADH2 ultimately reach molecular oxygen, and it involves a series of electron carriers which are arranged in the inner membrane of the mitochondria. The fractionalization of inner membrane revealed that there are 4 multimolecular complexes: complex 1, complex 2, complex 3, and complex 4. And also there are 2 mobile carriers of electrons. They are called ubiquinone and cytochrome C. And these ubiquinone and cytochrome C, they carry electrons from complexes to ultimately they take these electrons, transfer the electrons to elemental oxygen. So when all these occurs, for each, for each electron that moves from NADH2 to NADH2 to ubiquinone, there is a release of 2 protons. And when when FADH2 gets oxidized to FAD, 2 electrons make in they are taken up by ubiquinone, and ultimately from multi-complex 1 and also from 2, ultimately electrons will be reaching complex 3. This ubiquinone, a mobile carrier, takes electrons from NADH2 and also FADH2 and transfers them to cytochrome BC, which is otherwise called as complex 3. So from complex 3, the electrons will be carried to complex 4, which is nothing but cytochrome BC complex, and the transfer between cytochrome complex 3, complex 4 is facilitated by another mobile electron carriers called cytochromes. So ultimately from complex 4, the electrons are carried to elemental oxygen. So when protons move, the protons, when electrons are moving from complex 1, 2 protons are entering into a mitochondrial place. When electrons from FADH2 move to complex 3, 4 protons are released into intermitochondrial place. And when electrons are reaching the ultimate oxygen, 2 more protons are seen moving to intermitochondrial place. So starting from NADH2, you see that ultimately 8 protons are released. So these protons, with this oxidation, there is a lot of accumulation of protons. So I told you already, there is an accumulation of protons in the intermitochondrial place, which is 1,000 times more than their concentration in the what we call mitochondrial matrix. So so they like to move down their gradient. So this will be facilitated by, I told you, what we call ATP synthase, or otherwise called elementary particle, or F0, F1 particle. So for the energy that is present in 3 protons is sufficient to synthesize 1 ADP into ATP. So this conversion of ADP into ATP is called phosphorylation. As this phosphorylation is taking place with the oxidation, hence we call this as a oxidative phosphorylation. So so at the end, if you work out, what is happening. So in glycolysis, there is a production of what we call 2 NADH2s and 2 ATPs. So this is equivalent to what we call 6 ATPs. And in Krebs cycle, there is a synthesis of 8 NADH2s and FADH2s. From each NADH2, you get 3. So that way, you get 24 ATPs. And from 2 FADs, we get 4 ATPs. And 2 GTPs we are getting. So like that, so when you combine all these, from each glucose molecule at the end of electron transport system, we get a net of 36 ATPs. So this, if you work out in the form of, kilocalories, it accounts only for 40%. So 60% of the energy that is present. So it accounts to only 242 of the 686, only 242 kilocalories is available for the organism. Rest, 60% is evolved in the form of heat. So this is what we call, aerobic. So in the absence of oxygen, I told you glycolysis is common. Glucose breaks down to 2 molecules of pyruvic acid. Pyruvic acid in the presence of oxygen makes an entry into the mitochondrial matrix. There, decarboxylation, dehydrogenation takes place in the Krebs cycle. Ultimately, NADH2 are produced, FADH2 are produced. From that, you get, of course, we studied electron transport system. Ultimately, we get oxidative phosphorylation. Now, if oxygen is not available, what is the fate of pyruvic acid? In the absence of oxygen, pyruvic acid, changes to what we call this breakdown of pyruvic acid in the absence of oxygen is called anaerobic respiration. And this anaerobic respiration is otherwise we called fermentation. So the fermentation is nothing but literally a chemical change followed by effervescence. So here what happens based on the end product of fermentation? The fermentation is of 3 or 4 types: alcoholic fermentation, citric acid fermentation, oxalic acid fermentation, and what we call lactic acid fermentation. But in syllabus, we have only 2 fermentations. One is alcoholic fermentation. The other is, lactic acid. In alcoholic fermentation, it occurs in 2 steps. Generally, it was Louis Pasteur who first, worked out the process of fermentation, the reason for fermentation. He said that the yeast, if the yeast are introduced into sugar solution, they ultimately yeast converts the sugar into alcohol and also carbon dioxide. This was first discovered by Pasteur, but of course the reason behind this was worked out by Edward Buchner. He isolated zymase enzyme from yeast, and he gave the details of this fermentation. So this fermentation occurs in 2 steps. In the first step, the pyruvic acid gets decarboxylated to what we call acetaldehyde. Acetaldehyde in the presence of NADH2 gives rise to what we call, in the presence of what we call alcoholic dehydrogenase, it produces ethyl alcohol. So pyruvic acid in the first step decarboxylates into acetaldehyde, a 2-carbon compound. This pyruvic acid gives acetaldehyde. Acetaldehyde ultimately changes to ethyl alcohol, and for this an enzyme called alcoholic dehydrogenase is very much needed. So this is a 2-step fermentation. Is a 2-step reaction. In the first step, pyruvic acid changes to acetaldehyde. Acetaldehyde in the next step changes to ethyl alcohol. And the lactic acid fermentation is a fermentation wherein the lactic acid directly changes to, in the presence of lactic acid dehydrogenase, it changes to lactic acid. So based on the end product of fermentation, fermentation is alcoholic fermentation and lactic acid fermentation. So this is about the respiration. And as far as examination is concerned, this is very, very important. You may be asked, give in details of what we call Krebs cycle or electron transport system, or they may ask you write in detail about glycolysis and who are the people who elucidated this glycolysis, or what are the end products of glycolysis, etc. They may also ask you about what is fermentation, and write briefly about fermentation and about the importance of end products of fermentation. So at the end of fermentation, you get two important byproducts. One is alcohol. This is very much used in distilleries, and carbon dioxide that is produced will be used in bakeries. So these two end products, carbon dioxide and alcohol, are used in distilleries and also in bakeries. All our economy is depending on only this ethyl alcohol. All our liquor policies, all our liquor, or the income for the governments nowadays getting from this alcohol or the fermentation. For this, what is needed? The important organism is our yeast. So if you have any question as such, you may raise questions. If there is no question, so I'll call it a day. Tomorrow we'll meet same time from 3 to 4. Thank you.